Well, welcome once more. It gives me a great pleasure to introduce today's seminar speaker, Tim Stasevich from Colorado State University. Tim is Dean and Pingping Tsao Professor of Biochemistry at the Colorado State University. He's also a Monfort Professor and a Boettcher investigator. He is at the Department of Biochemistry and Molecular Biology. So I think the team started his independent career as an assistant professor less than 10 years ago now. And what was remarkable is that he moved through the ranks exceedingly fast. I think within a year, he moved from an associate to full professor. Tim did his PhD in the University of Maryland. With Dr. Einstein. Then he did his first postdoc at the further down the road at NCI at NIH, where he did some beautiful work on quantitative fluorescence microscopy and looked at protein dynamic binding, protein DNA binding dynamics in single cells with Professor McNally. And then he moved to Japan, where he did some clearly pioneering work before he moved back to the US to start his independent career. And Tim is doing some phenomenal work in using state-of-the-art fluorescent microscopy to look at how genes can be turned on and off in living cells or living organisms. And he has published some remarkable seminal work over the years. Recently, there was some beautiful work on capturing RNA polymerase transcription cycles at single copy gene that they monitor in a living cell. And that ability to resolve the spatial and temporal dynamics at the single gene level was really remarkable. So I'm personally really looking forward to hearing his talk today. And with no further ado, I want to pass the mic to Tim and tell us more about reading his title, moving the windows, imaging single-mRNA translation live. Tim, you're live.
All right, thank you, Markos. I really appreciate the chance and opportunity to speak here. I only wish it was in person. Markos didn't mention that I actually grew up not far from Ann Arbor in Canton, Michigan. Many of you Ann Arborites might not know where Canton is, but it's the place where the IKEA is. So it's the nearby IKEA. And that's where I got this chair, in fact. Yeah, so today I'm going to tell you a little bit about the work in my lab to image gene expression live and at the single molecule level. We have been looking at both transcription and translation, and today I'm going to give you more of a broad overview of our work to image single RNA translation. So if I were to kind of categorize my lab in one catchphrase, I'd say that we're basically creating gene light bulb technology. And what I mean by that is we're just trying to create technology to light up when a gene is on or off. And for this, we combine three strengths. We use live cell super-res microscopy. We develop novel protein tags, and we use computational modeling. We put all these three together to try to quantify single gene expression live. And so broadly speaking, we're interested in quantifying the central dogma of molecular biology, which, as many of you know, Francis Crick first stated in 1956, and that is that DNA makes RNA makes protein. Of course, there are lots of exceptions to this rule, but broadly speaking, this describes how your genotype gets converted into a phenotype. And these days, really, it's become possible to visualize this process at the single molecule level in living cells and organisms. And so this cell here is actually just one taken in our lab. You can see the nucleus here in red and the cytoplasm. It's a pretty large cell. This is actually an epithelial, a retinal RPE cell, which is quite flat and nice for imaging and quite big. And really, there are ways to monitor DNA, RNA, and protein all at the single molecule level. DNA, maybe you might be most familiar with because recently people have used CRISPR-Cas9 or deactivated Cas9 to light up individual genes in the cell. We often use the MS2 system, which was developed in the Singer lab, to light up individual RNA. And in fact, in this cell here, all these little green and blue spots are individual RNA molecules. And you can see their dynamic is quite distinct when they're close to the nucleus here compared to out here in the cytoplasm. The coloring here goes from kind of this greenish color to this bluish color. That designates the depth in the Z space, in the direction perpendicular to the screen. And so you can see these RNA that are out here right on top of the nucleus, they're kind of immobilized. They're probably coming out of the nucleus. And then when they come out into the cytoplasm to be translated, they can be quite mobile. And here at the edge, they're very mobile. It's also possible to image proteins at the single molecule level. For this, the HILO tag has become quite popular, and people have looked at transcription factors. In fact, here we have RNA polymerase in red. And so you can actually see the nucleus and you can see where the polymerase is. Some of these brighter spots are probably bright transcription zones. And then this little spot right here, I want to draw your attention to, that's probably a transcription site, so where the RNA is being created. And then ultimately it leaves the site, gets exported out here into the cytoplasm and translated. Now, of course, there are ways to image the different processes of the central dogma as well. Transcription you can quantify by doing, for example, fluorescence recovery after photobleaching experiments, where you might take this little transcription site and photobleach it so the fluorophores turn off, and then you can watch the fluorescence return back to normal. That's going to give you some information about how fast transcription is occurring. One of the last aspects of the central dogma to be visualized at the single molecule level was translation. And so today I want to tell you about why basically it was hard to image translation and how our lab has and others have helped develop technology so that you can actually visualize this process now at the single molecule level. Okay, so why is it hard to see translation? And this cartoon kind of summarizes the point right here. So here I have an RNA and it's being translated by a ribosome. And as it's being translated, a nascent polypeptide chain is elongating, coming out the ribosomal exit tunnel, and it elongates. And eventually it codes for or creates a protein of interest, a POI. And even if you take a green fluorescent protein tag and you put it on the end terminus of this POI, so that it's the very first thing to be translated, by the time this green fluorescent protein will mature and light up, translation is long over. And so if you're using a green fluorescent protein to light up translation, you're not going to be able to see it just because it takes too long for that GFP to light up and to mature itself. And so this is basically a fundamental problem. Now, people have used GFP again to monitor proteins of interest in cells and see where they're going. But unfortunately, whenever you're looking at a cell that has GFP fluorescence, all you're seeing are the mature proteins. All of the nascent proteins that are still maybe being translated, you can't see. So that's an important point to keep in mind. So our idea when I started my lab at Colorado State University was to, instead of have the fluorescence form at the site of translation, maybe instead we could recruit preformed fluorescence to the site of translation as it's occurring. And for this, we thought we could do something analogous to how the MS2 system lights up RNA. And that is, we'd have an intrabody that would bind to a very short peptide epitope, something like the flag tag, which is just 9 amino acids and doesn't have any structure. And so if you put a bunch of flag tags on the end terminus of your protein of interest, that tag will emerge from the ribosomal exit tunnel, and it doesn't need to fold before it can be bound by a fluorescent intrabody. And if you have enough flag tags on the end of the protein of interest, you can amplify the signal. Furthermore, you get another level of amplification just because a single RNA can actually be translated by more than one ribosome at a time. So we thought maybe we could use this system to get double amplification and actually see the site of translation. And so I started this project with Tatsuya Morisaki. He came with me to Colorado State University, and our first graduate student, Ken Lyon, shown here. And so the system that we developed is basically what I showed you. You have a 10x flag tag on the N terminus of your protein of interest. And then in the 3' UTR of the gene, we put a 24x MS2 tag. This is basically a tag that we can light up the RNA with. So when transcription occurs, this RNA tag forms these stem loops, and these stem loops are bound by a fluorescent MS2 coprotein. So once the RNA is processed and exported from the nucleus, you can actually see the RNA lit up in purple here with this MS2 RNA tag. Then when translation occurs, again, we're going to recruit these flag intrabodies to the translation site. And so when we see basically the co-movement and co-localization of our green translation tag and our purple RNA tag, then we can be sure that we're actually imaging translation. And so when everything goes right, this can look really nice inside of a living cell. Here's actually an example, and this is a movie. What this actually is a maximum intensity projection. So what we've done is we've imaged the entire cell, the whole volume, once every second. And then we take the maximum intensity pixel from one of the planes for every single pixel. And by doing that, all of these little spots that you see moving around, which are the RNA, they actually always stay in focus, because no matter what plane they're going to go into, they're going to be the maximum intensity, unless, of course, they cross over one another. But what you're basically looking at here is RNA is in purple, the translation signal is in green, and what you see is a lot of little white spots. And these white spots are basically just the co-localization of that green translation signal and the purple RNA signal. And you can see most of the RNA in this case are actually being translated, but this one here is not. There's also another one up there that's not really being translated. But most of them have that green signal that's co-moving with them. Now, once the protein is translated, in this particular case, we actually made a nuclear protein as our protein of interest. All of the green mature protein goes into the nucleus here. So that's why the nucleus appears green. All the mature protein goes there. So if you were imaging this particular protein of interest with the green fluorescent protein, all you would see is this nuclear signal. Now, we also have this haze of green, kind of a cloud of green fluorescence in the background. That's just the fluorescent green intrabodies that bind to the flag epitope tag. They're diffusing all over, but only at the site of translation are they significantly amplified above the background signal. And so you can actually use that to actually see translation. So hopefully this is all clear for everyone. It's pretty straightforward technology, but using it, we're able to track not only the RNA, but also the translation status. Now, when we first developed this technology, one of the first things I wanted to do for my own sanity was to make sure that we were really imaging translation. For this, of course, you'd want to add a translational inhibitor. And puromycin is a perfect inhibitor for our experiments because it rapidly knocks the nascent chain, the nascent polypeptide chain, right off of the RNA by splitting apart the ribosome. And so here's a cell where you can see lots of little white translation sites. When I play the movie, what's going to happen is those white sites are going to turn purple only. And that's because translation is being turned off. So I'll play the movie. It's really fast, actually. So right now that you can see they're all purple, essentially. I'll play it one more time. And if you were to zoom in on one of those RNA, what you would see is the RNA signal persists through time, but the protein signal after you add puromycin just kind of slowly goes away. Well, actually relatively quickly. And in the control example, of course, both signals will persist through time. And so this was the experiment that really convinced me that yes, indeed, we were really imaging translation. Not only were these spots of fluorescence co-moving with RNA, but they were also responding to translational inhibitors, as you would expect. Okay, so before I go a little bit further, I just wanted to give you guys all a broad background on kind of our system. And for me, I'm actually come from a background in physics, and microscopes are a big part of our lab. So I thought I'd spend a little bit of time talking about how we actually image these things. So for all of the experiments that or all of the movies that I'm going to show you in this talk today, they were all done using this technique called HILO, which stands for highly inclined laminated optical sheet microscopy. And this was actually developed by Makio Tokunaga, who's at Tokyo Tech. And back when I was doing my postdoc in Japan, I got to actually work with Makio Tokunaga and check out his microscope. He had one of the only 3-color single molecule microscopes in the world for imaging in thicker specimens. And so actually, the way this works, it's sometimes referred to as dirty turf because it's using objective-based turf, which is another technique that Makio Tokunaga developed, but then you go slightly off so that the angle of the light is not in turf mode, but comes in at a very sharp angle to the cell. And the thickness of this light sheet is about 3 microns about. And so when you're talking about a mammalian cell nucleus, which can be 10 microns in diameter or in thickness, this is kind of a slice of that. And because you're only illuminating a slice of the cell, you get increased signal to noise. And so if you just do normal epifluorescence where you're illuminating everything from below, your signal will look like this. But if you go into HILO mode, you get about a factor of 8 improvement in the signal to noise. And for single molecule tracking and for our experiments, this factor of 8 is really critical because it allows us to turn the lasers down and track for 8 or 10 times longer than we would be able to otherwise. So when I came back from Japan, I was at Genelia Research Campus, and there I met Brian English, who had made a very similar setup based on HILO. But what I liked about his setup is he had an open microscope system, so you could really change anything you wanted. And so what we did is his system is shown here on the right. We basically replicated it at Colorado State. But we did it with 2 cameras instead of 3, which they had at Genelia at that time. But our microscope, we call it FIXle for fun, because there are a lot of FIXle bikes in Fort Collins, Colorado, but also because it's a single gear kind of microscope. It does one thing and it does it really well, and that's multicolor single molecule tracking. It has a fixed pixel size, optimal filter set for 3 colors, and everything is customizable, accessible, and it's completely open source. So we use Micromanager and Arduino to control the microscope. And we basically have 3 laser lines for lighting up red, green, and blue proteins. We have a 4th laser for UV for doing photo bleaching or optogenetic photoactivation. Our red emission path goes to one camera, and then our green and our blue emission paths go to the other camera. So these 2 cameras we call CAM and RAM, which for those who aren't familiar with CSU, these are mascot, CAM and RAM. Okay, so we also are kind of cheapskates. We wanted to spend a lot of money on our cameras, but we wanted to save money elsewhere. And for that, we used kind of do-it-yourself open source technology. And the way this works is we have Micromanager, which is open source software, sends a signal to one of our cameras, and that signal then goes to a microcontroller, an Arduino $20 microcontroller, and then that will split the signal to our different lasers so that we can image in different colors or move the stage in different colors whenever we want. And basically, this just saved us a tremendous amount of money so that we could focus all of our money on the important camera component. We also made our own custom stage top incubator. These things can cost, $15,000 to $20,000. The one we built cost $200, and we used a fish tank temperature controller, actually, which you can find the link for here. But anyways, as you can tell, we like to kind of do things ourselves, and so our microscope is quite unique. Okay, so what can we quantify with our system? Well, it turns out that this way of imaging translation is highly quantifiable. Of course, you can look at the mobility of translation sites or their localization, see where translation is occurring in a cell. Is it occurring at the nuclear membrane, or maybe it's out at the edge, or maybe it's at the mitochondria, right? So we can quantify all of those things. If we look closely and do some super res microscopy, we can actually also say something about the general shape of a translation site or a polysome. Are they stretched out or are they more globular? So far, we mainly see kind of globular sorts of shapes. We rarely see long stretched out translation sites. And then of course, zooming in, because the fluorescence intensity is going to be proportional to the number of nascent polypeptide chains that are associated with the RNA, and that's directly proportional to the number of ribosomes that are moving along the RNA, our fluorescent signal is actually going to tell us something about translation. So the brightness is going to get brighter if there's more ribosomes, and as another one initiates, you'll get a fluctuation up in the fluorescence, and as they terminate, the fluorescence is going to fluctuate down. So we can use all of this information to quantify things like the density of ribosomes along the transcript, the rate of elongation, as well as the rate of initiation. And just to drive this point home further, I'll say there's actually 4 ways to measure the elongation rate using our setup. So the first way is kind of illustrated here. If you imagine a transcript where it's translated very infrequently, so that at any given time there might only be one ribosome moving along it. So here at the top I have the transcript. This would be the start codon, the stop codon, and these little green squares represent the flag tags. And these are the short epitopes that our fluorescent interbodies are going to bind to when they're translated. And so there are 10 of them. And as the ribosome moves across that portion of the tag region, the fluorescence is going to go up because the flag tags are going to emerge from the ribosomal exit tunnel and recruit fluorescent interbodies to the nascent polypeptide chain. Then when the ribosome finishes translating the tagged portion, the fluorescence is just going to remain constant as it moves across the rest of the gene. And then eventually when it hits the stop codon, the fluorescence is going to completely drop. And so this represents kind of an idealized fluorescent signal you would get from a single RNA. But of course, most RNA are not translated by single ribosomes. Most RNA that we've observed, well, and this depends a little bit on the length and size of the RNA, but most of them are translated in polysomes by more than one ribosome at a time. And so in this case, it can be a little bit more tricky to actually get the elongation rate. On the left, you can just kind of measure how long it takes this signal to appear and then disappear. That's divide that time by the length of the transcript, you get the elongation rate. But when you have a lot of RNA on your, or a lot of ribosomes on your RNA, this fluorescence can fluctuate up and down. And so in this scenario, which is probably more common, what we do is fluorescence correlation spectroscopy. You can ask yourself, how long does the fluorescence remain correlated with itself in the future? And when that correlation time, that probability of being correlated with yourself a later time in the future, when that goes to 0, that essentially gives you the elongation time as well. 2 other ways to measure the elongation rate. You can do a drug assay. Herringtonin basically inhibits translation initiation. So after you add herringtonin to your cell, the ribosomes just run off the transcript one by one. So herringtonin doesn't disturb elongation or termination. It only inhibits initiation. So what happens is all of your ribosomes that were there, one by one, they just eventually run off the transcript. And by the time the last one comes off, that gives you a rough estimate for the elongation time as well. Similarly, we can do a photobleaching experiment where we would photobleach our translating RNA so that at time 2 here, most of the fluorescence is off, but as new ribosomes begin to initiate again, the fluorescence will recover back to what it was. And again, the time it takes for the fluorescence to recover gives you an estimate of the elongation rate. So I hope this basically gives you a a better picture of how we can actually quantify the fluorescence in these sorts of experiments. For those who are not so familiar with fluorescence correlation spectroscopy, I thought I'd maybe just illustrate how these fluorescence fluctuations give you information about the transcript you're looking at. So at the top here, we have a long gene, which is going to be a long protein of interest. And at the bottom, we have a short transcript with a short protein of interest. And everything else is exactly the same in these illustrations, but what you can see is that the fluorescence of the signal that you would measure at this particular polysome are quite distinct. So the long transcript has these really long fluctuations, whereas the short transcript, as you might expect, has shorter fluctuations. And so for example, here, as you watch the fluorescence come in, this long transcript can suddenly get loaded by a bunch of ribosomes and the fluorescence gets really bright, right? And then eventually, as time goes on, that fluorescence is going to drop and eventually go to zero. That size of that kind of peak tells you roughly how long translation takes, whereas these little peaks here tell you roughly how long translation takes for the shorter transcript. And again, if you just ask yourself, well, what's the probability of seeing the same fluorescence sometime t into the future? For the short gene, that probability goes to zero very quickly, but for the long gene, that probability goes away more slowly. Just because you're going to see the same ribosome there sometime later into the future, when for the short gene, the ribosome would already be gone. So if you just look at this probability that, or this autocorrelation function for different genes of different length, that's exactly what we see. So for a really long gene, which is 1,500 amino acids, this probability goes away there. But for a very short gene that's just, say, 300 amino acids, it drops right there. If you take those probability, the time of when the autocorrelation goes to zero, these times here, here, and here, and just divide by the length of the transcript, you can get the translation elongation rate. And what we found is for multiple different length transcripts, essentially it was always somewhere around 10 amino acids per second. Okay, so we've also generalize this approach. So we were looking at the flag tag where you're looking at translation in green. We can use other epitope tags to light up translation in other colors. The HA tag is another short amino acid. I think it's 9 amino acids as well. It can serve just like the flag tag, and there are intrabodies that you can use to light up these nascent chains in blue so that you can track 2 different transcripts in 2 different colors. And this just shows a cell where we have all of the RNA here in red zipping around, and some of them have blue fluorescence associated with them, other ones have green. That depends on which one you're seeing, flag tag or HA tag fluorescence. And so in this way, we can actually monitor 2 transcripts at the same time in a single cell translation of them both. One of the surprising things we found was that there were some spots where we saw all 3 colors in the same spot, which would suggest this translation is occurring kind of in a higher order structure where you have 2 polysomes coming together to be translated together. And that's quite interesting because you can imagine that there would be circumstances where you'd want 2 transcripts to be regulated in synchrony. And maybe when they come together, the local concentration of translation factors is high, so the probability of translation of both of them goes up. At least that's our thought right now. Anyways, so this is what kind of started our whole lab. And since this time, we've just been having a lot of fun imaging translation in lots of different colors and having all sorts of cool applications. And one of the nice things about when we published this first paper is there were a group of other papers that came out at almost the same time who used the sun tag to do a very similar sort of translation measurement. And it was very gratifying to see that essentially we were all measuring elongation rates that were on the order of between 3 and 20 amino acids per second. And then our initiation rates that we were measuring was roughly 1 ribosome coming in every 15 to 30 seconds. And that was over a number of different transcripts. It's kind of interesting how similar these numbers are. Also, if you looked at the density of ribosomes per nucleotide, That ranged from one ribosome every 200 nucleotides all the way up to 800 or 900 nucleotides. And so, yeah, I think this technique is very quantitative, and there's lots of information that you can pull from it. Okay, again, since then we've done lots of cool things, and these are all little movies. I'm sure many of you here at the University of Michigan are aware of this story, looking at translation shutdown during stress, which was from Stephanie, who's now at University of Michigan. Going to do some really cool things, I'm sure. But what we were able to show in that paper was that translation seems to be shut down essentially outside of the stress granule. And then only once the translation is shut down outside of the granule do you see stable interaction of the RNA with the stress granule. With Ken Lyon, my first graduate student who about a year ago, he graduated, we looked at frame shifting, translational frame shifting, where you can have obviously two different frames being translated at the same time on a single transcript. And this process of frame shifting is used by viruses quite often. The quintessential example is GAG-POL in HIV. And so we actually looked at that HIV frame shift sequence. And the surprising thing we saw is that there were a few RNA that were actually frame shifting, but when they did frame shift, they were strongly frame shifting. It was like bursts of frame shifting that we saw on a subset of RNA. And so that's going to be interesting, I think, to follow up on. We've also developed some genetically encoded intrabodies to light up our translation. I'll talk a little bit more about that in a second. That's actually Ning Tsao. She just was awarded a K99 to continue to pursue this sort of probe development and look at co-translational folding dynamics, actually. And then most recently, we had our paper looking at iris versus cap translation initiation. So viruses use internal ribosomal entry sites to initiate translation, and we created a biosensor that allowed us to compare in two different colors whether translation's initiated in an iris-dependent manner or in a cap-dependent manner. And so that was from Amanda Cook, who just graduated actually this past summer. And then I guess I should also mention we have this new tethering reporter where we're actually tethering different molecules to translating RNAs to see the impact of those tethered molecules on the translation dynamic. And so we started with AGO2, which is part of the micro-RISC machinery, which microRNA to target specific RNA and shut off translation. And so this is currently actually on bioRxiv. Okay, so I'm not going to actually go into details on any of these. They're all available. You can read about them. What I wanted to spend the last 10 minutes or so of my talk discussing is some of the new stuff that we're doing that hasn't yet been published. And so kind of what we have on the horizon. And so part of that is just developing more of these genetically encoded intrabodies to light up short peptide epitopes in living cells, right? So Ning Tsao basically has been developing these, and what we wanted to do is to develop one that could bind the HA tag, the flag tag, all of those standard epitope tags that are just being used everywhere in biotechnology. We thought, wouldn't it be nice to have a genetically encoded intrabody version of that tag? And it turns out that intrabodies are actually pretty hard to develop. And that's because when you think about an antibody, they really weren't meant to be expressed inside of a cell. They're meant to be excreted from cells and expressed in the bloodstream. And so putting an antibody back into a cell where the pH is a little bit different, it's a reducing environment, what tends to happen is the disulfide bonds in the antibody break apart. And so unless you have a really stable fold, keeping an antibody folded inside of a living cell is challenging. And so the way this generally works, people, also I should mention antibodies are just big, they're multi-chained, right? So what people tend to do is instead of using full-length antibodies is they'll use shorter versions. And the common one that is used is the SCFV format, which stands for single chain variable fragment. And that's basically just the variable regions of a full-length antibody that you then combine with a linker. And this linker, the way it's set up, it's got to go from N terminal to C terminal. That ends up forcing the linker to kind of have to span in this weird sort of architecture. And so getting an SCFV to actually fold inside of a living cell turns out to be a big challenge. And so what Ning has been basically doing is creating what we call Frankenbodies, which are chimeric SCFV. And so what you do is you take a really stable SCFV that we know folds and can be expressed inside of a living cell, and you just take the 6 CDRs, the complementary determining regions from the antibody, and you graft them onto that stable scaffold. And by doing that, she was able to make an HA version. So this just shows the RNA. And here we're lighting up translation with the HA tag. But more recently, she's made a flag tag version. This actually just came out in Genes to Cells, and it's now, both of these SCFV are available on Adgene. So if you're interested in lighting up HA tags or flag tags in your cells, visualizing where those tags are, you can go to Adgene and purchase GFP tag versions of each of these interbodies. And more recently, we have a new one we call the U tag, which is a super tight binder. And so we actually looked at the binding strength of our interbodies to each of their respective tag, whether it be the HA tag, the flag tag, or the U tag. All of these tags are just about, they're each 8 or 9 amino acids in length. And here just shows you the binding recovery curve. So we have intensity versus time, and we photo bleach. And the flag tag turns over pretty rapidly in about 100 seconds. Most of the flag tag interbodies are going to unbind and be replaced. The HA tag is somewhere in the middle in about 200 or 300 seconds in purple. That will recover. But this U tag is just a super tight binder. Basically, it'll remain bound to its epitope for 500 or 600 seconds at a time. And so really you can use these, depending on your application, it might be actually useful to have an interbody that binds really tightly like this U tag, or it might be useful to have a weaker binder like the flag version. So we think this will be kind of nice because you can kind of tune the binding strength by just choosing which epitope system you want to use. And just to show you, here's what translation looks like with our HA interbody. And then when you add puromycin to inhibit translation, watch all these spots just kind of fade out. And that occurs really within a minute to 2 minutes. This fading out occurs. Okay, another thing that we've started to do, looking at my time, where are we? Looks like we still have a little bit more time. Another thing we're starting to explore is translation plus optogenetics. And for this, we have an assay where we can actually cluster mRNA. And this is done through GFP, actually. And so we have an RNA that is labeled in purple with the MS2 tag, and we have another tag in blue here, and then translations monitored in green. And then we can shine a UV laser, and it causes all these blue tags to cluster together. So here is just a movie where you can see green and blue together. So here's a translation site. Unfortunately, even at this time 0, there's a little bit of clustering, but when I play the movie, all of the GFP is going to cluster, which is shown in blue here. It's all going to cluster together. And this translation site, watch it, you're going to see what happens to translation when you cluster a bunch of RNA together. So you can actually see that signal of that particular RNA decays with time. But I'll just show, just by looking at the translation signal, you can see it just kind of fades out. And what we think is happening there is when we're just clustering all those RNA together, this is kind of like a synthetic granule. And we can ask ourselves, well, what happens to translation in a really crowded environment? And if it's too crowded, we think translation initiation can no longer occur. But interestingly, the ribosomes are still able to elongate. In fact, when we add in puromycin into these cells, even when the translation sites are clustered like that, they still respond to puromycin. So elongation is still occurring, but it seems like initiation may be inhibited. And not all RNA gets silenced, right? So this one here doesn't seem to be silenced, right? So we're trying to kind of figure out what are the biophysical rules that just from crowding, what is the contribution of that to silencing translation? So this will be kind of a fun system to explore. And then later we're planning on maybe putting like Argonaut, AGO2 with a GFP tag. We can cluster Argonaut with this translating RNA and see if translation goes way faster. Or we could really cluster any GFP tag protein with our translating RNA. And then last but not least, I just want to mention that we are also trying to take our tags and put them into endogenous loci using CRISPR-Cas9. And for this, actually O'Neill Wiggin in our lab has really pioneered this, because getting these big repetitive tags into the endogenous genome is difficult. And so what O'Neill did is he basically put a 24x MS2 tag along with Clover at this C-terminal end of the Myo2A locus, which is just the heavy chain of myosin. And so the beauty of this system is the myosin ends up with the Clover, so the mature myosin you can see in green, but it also has a 24x MS2 stem loop, so you can visualize transcription. And so just to give you an idea, here in green, you can see the mature myosin, and in red, you can see RNA, and these big bright spots that you're seeing in the nucleus are actually transcriptional bursts. So this is a 10-hour movie, and you can see that gene turning on and off in a burst-like fashion. And that took one round of CRISPR editing, but O'Neill, to his credit, this wasn't enough. He did a full second round of CRISPR editing with one of our undergraduates, and he put another tag on the N-terminus, which had a 6x flag tag. So you could actually visualize translation using flag. And this is a pretty cool system because you can see, even though you have all these tags on the heavy chain of myosin, it's still functional. It's still forming actomyosin fibers here. And you can see that red to green sort of placement. So the system is working really beautifully. But the advantage is now we can see the entire central dogma at the single molecule resolution, including transcription and translation. So here I'm just showing you in these cells a translation site. And you can see that the translation here in the flag is actually oscillating up and down, but the RNA is kind of remaining constitutive. And then the GFP shows the mature myosin. Notice you don't see that signal at the translation site. That's just providing more evidence that GFP takes a long time before it fluoresces. So even if you put it on a gene, you're not going to see it until later. And then here at the top, we're just looking at 2 RNAs that are being translated. So this is the flag channel, and you can see the translation sites. But the mature GFP channel, you don't actually see the fluorescence at those 2 sites yet. So that's, again, the power of our technology. Now we can actually see RNA, we can quantify transcription, we can count the number of RNA, we can count the number that are being translated. So we can really distinguish, for example, RNA decay from translational regulation just by counting RNA and actually counting how many are being translated. Here's just some more really beautiful images. I'll stop here, where you can see RNA that appear to be locally translated. So here's RNA. It's at the end of one of these filaments. And you can see translation occurring, but the mature GFP hasn't lit up yet. And likewise, here you can see 2 RNA that appear to be being translated right at the nuclear periphery. So this is quite interesting. We're trying to figure out why translation of myosin is occurring right here at the nuclear periphery. Does that have any biological significance? You can see those 2 spots right there, as well as from there. So it's a pretty common phenotype that we see. So with that, I think I will just conclude. So yeah, again, my dream is creating these light bulbs for imaging gene expression live. And I think we're getting closer, combining our microscopes with our new probe sets to light up translation in a rainbow of colors. Together with computational modeling and genome editing, I think we can really start to look at how gene networks are actually being regulated together. And so that's kind of where I'd like to go with this. And of course, I have to thank my lab, who really did all the work. You know, we are situated at Colorado State University, where we have mountains nearby. I think this is all us, the top of Horsetooth, for those who are familiar with the area, overlooking the reservoir. With that, I think I will end and just thank you all for your time.
Thank you, team. I wish you were here in present. Here are joyous clapping. Very, very interesting work. Tremendously visually arresting as well at the same time. I really personally enjoyed. So feel free to start posting questions on the Q&A or ask your questions. So I can either read your questions, or you're more than welcome to ask them live as well. Team, till we get the first question, I guess I can go ahead. Oh, still have one over here from Joe. Joe, do you want to ask your question, or do you want me to? Okay, so I'll go ahead and.
Using a standard turf microscope, are these methods still possible? Yeah, no, that's a great question. Yeah, so turf, we're essentially in dirty turf mode, right? So if your RNA, using a standard turf microscope, if the RNA are close enough to the cover slip, you'll be able to see them in turf, but you can always go into dirty turf mode, which is where we're operating. And it depends on how you're making turf, right? We are doing it with objective-based turf, which I think is the standard now. But certainly, if you have like a prism and you're doing turf from above, it might be a little, or turf from a different angle, it might be a little more difficult. I'm not really sure, but certainly there's no reason you couldn't do this with a standard turf microscope. You can also visualize translation with a confocal microscope. In fact, some of those last movies showing endogenous myosin were confocal images, just a standard Zeiss confocal. So nothing that special, because we're just amplifying the fluorescence so much. But good question.
We have an additional question for you on Q&A. So do you think that adding tags to the C or the N terminal of genes affect mRNA localization or any other properties?
Yes, absolutely, they can affect, just like GFP, right? I mean, you just can't put GFP anywhere. And in the end, you really got to test on a case-by-case basis. And so what we often will do, particularly for the myosin locus, what we did is fish. So you can fish the endogenous genes and see how many RNA you're getting per cell. And then we CRISPR edited our tags in, and then we fished against the MS2 tag, or you can just image the MS2 tag. And you want to make sure that the number of RNA is roughly the same per cell. But absolutely, you really just got to be careful, and on a case-by-case basis, check that all of these tags are not interfering. I certainly know of cases where if you put a big tag on, you can absolutely destabilize either the RNA or the protein. And so you just got to check.
Thank you.
And fish is the way to go.
We have a, oh, okay. Guoming, I'm going to go ahead and ask you a question since your mic is not working. So Guoming is asking, RNA translation and RNA diffusion can be happening at two different time scales. The translational imaging takes hours, while RNA diffusion could be very quick. How do you think we can bridge these two pieces of information?
Yeah, better fluorophores. But I mean, I'd love to image longer, right, without photo bleaching. So that's the biggest problem, is you're perturbing your system, right? And so a more sensitive microscope is going to allow you to put fewer tags, fewer perturbations, and image for longer. So obviously on the microscope end, if we can develop better cameras, that's better. If we can develop probes that are maybe light up, like one thing I hate about our movies, even though they're beautiful in many ways, is that green hazy background that we get from all the freely diffusing intrabodies. Wouldn't it be great if the intrabody only lit up when it's bound to the to its epitope? And there are people working on those sorts of things. They're called flash bodies. There's a guy at Tokyo Tech who's doing that as well. But again, that might affect the timing, but that would increase your signal to noise, and it would allow you to turn your lasers down or maybe track for longer without photo bleaching. And so, yeah, we need to be able to bridge those timescales. We need to be able to image for seconds over a period of hours. And yeah, I think that's the goal.
Cool. More questions are welcome. In the meantime, team, when you how much have you studied? Like, well, let me rephrase this. When it comes to the clustering experiments that you showed us, where you mentioned that you see no longer a translational initiation and you could still see elongation. Are the elongation rates impacted by this clustering? Is there something that you can control? Is that something that you can potentially start like thinking about the physiological significance of some of these clustering events in the cell and how they impact translation rates?
Exactly. We're really interested in how clustering into these synthetic organelles, you might say, or synthetic granules, how that, because it's a controlled environment, we can control what we put in and when it goes in. And so, I mean, the idea is it's kind of like doing biochemistry in a living cell, right? Where now this cluster is your little test tube, right? So that's kind of why I'm really excited about this technology. I feel like there's a lot of potential, but of course, just the act of clustering is going to perturb things. And so the very first thing we need to do is say, okay, yeah, how does it impact translation? And it clearly seems to impact it. According to our data, elongation isn't as perturbed as initiation. But to truly say that, we need to compare the Harringtonian type of experiment where we can shut off translation by completely blocking initiation, get the runoff rate in that scenario, and then do the same thing with just clustering, and we can see if it is a little bit longer or a little bit shorter. My guess is elongation is probably perturbed. It's probably a little slower, but not as much as I would have thought.
Do we have any more questions from the audience? Well, if not, then please join me in thanking team once more for joining us and for an awesome seminar. Thank you, team. Again, hopefully next time we'll see you in person over here. Well, thank you all for coming.
Yeah, thank you.